Okay, what's up? I just tried to make a video twice on my old computer. It's got a couple of non-COVID viruses, and so it didn't work. So I'm going to try it on this one. Okay, so chapter 11, okay, as I've explained to the computer a couple of times, is the cell communication chapter. Now, the cool thing about cell communication is that it's ubiquitous. It applies to every way that every cell communicates. So this is plants, animals. This is your immune system versus your digestive system versus your nervous system, as well as bacteria, right? Okay, to some extent. I guess I can't say that for sure, but certainly they use similar pathways because these are just kind of throughout microbiology. Okay, so I think if I could just take a second to explain a metaphor here. This is like teaching someone how to garden. Okay, so if I was trying to teach you how to garden, I wouldn't teach you how to grow tomatoes. Okay, I would teach you how to just grow plants in general. I'll be like, okay, so we need to have, manure, fertilizer in the soil. You need to have mulch on top. You need to put a seed in there. You should start the seed inside and then transplant it outdoors. You need to have sunlight. You need to have water. You need to have a watering schedule. It needs to have the right amount of sun. It needs to be in the right temperature. You need to have it in the right spot in your yard, right? So like if I taught you the general principles of how to grow something, then you could grow tomatoes, eggplant, basil, zucchini, and peppers, all like I'm growing right now, okay? But it doesn't give you the specifics of any individual one, right? So chapter 11 in cell communication is giving you a general idea of how all cells communicate. So it's not going to tell you how like your neurons communicate specifically, but it will tell you the general mechanisms that they use in order to receive signals and actually not really send signals, but this is mostly like signal reception and then causing a change inside of a cell. Okay, so hopefully that framework helps a little bit. So there's a bunch of acronyms, and the biggest thing to understand with acronyms is that they're not scary. You just need to memorize them and realize that there's a lot of repetition. And the other thing, whenever we're talking about pathways and microbiology, is you need to know what the size of things are and what category they fall under. So whether it is a molecule versus a protein versus a process is very important. So this ties directly into cell respiration and photosynthesis as a unit, which is one that just has processes, molecules, and what was the other one? Proteins, okay? And some of the proteins are enzymatic, meaning they do work, and other proteins are just structural. Now, fortunately, there's not really any structural proteins here. They're pretty much all enzymatic, meaning they're going to end in ACE, and they're going to do some sort of work, okay? So let's define all the acronyms, and then let's talk about, what's molecules and proteins and whatnot, okay? So cAMP is pretty much, to me, like the central idea behind this. If you understand what cAMP is, then everything else kind of like ties into that, okay? So cAMP is cyclic AMP. And cyclic, meaning it's literally just in a circle, and AMP stands for adenosine monophosphate, okay? Sounds pretty similar to adenosine diphosphate or adenosine triphosphate, are charged and uncharged versions of a battery that is made from the RNA adenine along with a phosphate on it. And I believe ribose is present there as well, okay? So if you have one phosphate, it's AMP. If you have two phosphates, it's ADP. If you have three phosphates, it's ATP, right? So don't be scared about this molecule. Cyclic AMP is just one phosphate, and it's kind of bound into like some sort of more rounded circular thing. Don't really worry about that, okay? But it's a molecule, okay? So this molecule is a signaling molecule, okay, that activates pKa, or sometimes they'll say protein kinase. Now, kinase, ends in ACE, means an enzyme. Kine means movement. So it's just an enzyme that does something, okay? It's just going to move something around and convert something. It's called a protein kinase because it's going to be working with proteins, doing something with proteins, right? It's not called a protein kinase because it is a protein, because of course if it ends in ACE, it's an enzyme when you know it's already a protein. So if it's a protein kinase, it's a protein that works on other proteins. Nice. So in order to activate protein kinase A or protein kinase B or just a protein kinase, anytime you see anything PK, pKa, whatever, that is going to be activated by the molecule cAMP, cyclic AMP. Now, how do you activate cAMP? Well, it's no surprise that you are going to get cAMP from ATP, right? You're going to have to chop off 2 of the phosphates, and then it's going to go and it's going to bind to a activation site, or perhaps an allosteric activation site, as an activator to pKa, so that it can do some work. Okay, now keep looking left to my notes here, just so you know. Not like I'm crazy, because that's definitely not true. So, okay, so how do we activate cyclic AMP so that it can activate protein kinase, and then the protein kinase can like work on any protein, can basically just impact it. Like I said, there's general applications here. It could impact something in the nucleus. It could impact something in the ribosome, something in the rough ER, somewhere throughout the cell. Typically, though, we're talking about impacting the nucleus, impacting the protein synthesis pathway, so that we can make a protein to respond to something, okay? And what we are responding to is a signal received from outside of the cell. Now, a signal coming from outside of the cell would have to either go in through the phospholipid bilayer, or it would, in this case, have to touch something on the phospholipid bilayer, which would then change shape, and then the change of shape would cause a reaction to happen on the inside of the cell, which would then cause a reaction to happen, and then another reaction, then another reaction, then another reaction, then another reaction. So this is like a total domino effect, okay? And the start of the domino effect is what I just described in terms of cAMP being converted from ATP into cAMP, and then docking onto a PK protein kinase, and then continuing the signal pathway, okay? So how do we get cAMP to be made from ATP? A ligand, which is a molecule of some sort, not a protein, smaller than a protein. A ligand molecule is going to bind to one of the examples I'm seeing, and I think it's pretty ubiquitous again, is the G proteins, okay? And if you see something like GPC or GPCR or PCR, and there's a little G next to it, all of those things are the same thing. It's a protein complex receiver, and it works with G proteins, okay? So the protein complex is something that's stuck in the phospholipid bilayer, and it has a little docking site, what are those called? Just a little spot where something docks. I forget the name of it right now. Okay, it's not an allosteric site. It might just be called an active site. It might be, yeah, anyway. So the ligand comes and it binds to that little spot, okay, on the G protein complex receiver or receptor. It binds there, and then that changes the shape of the GPCR. Now the GPCR is touching a G protein. Now, the G protein is made of 3 things: alpha, beta, and gamma. Gamma looks like a Y. Alpha and beta, you should know, okay? So those 3 parts are the G protein, and they are bound to the GPCR, and they're on the inside of the cell. So the ligand comes and binds right here, and then that changes the shape, and then these guys right here are going to split apart. One of them is going to stay, that is the alpha component, and then the beta and the gamma component are going to float away, okay? Now, in this process, GDP is converted into GTP, okay? All of that thing, when this binds, this changes shape, these break away, GDP is converted into GTP, and the GTP stays with the alpha section, okay? Whereas the beta and the gamma part are going to split apart, and they're going to go dock with another protein, which will do something. The alpha and the ATP or GTP is going to potentially dock to a different protein, or it's going to stay right there with the GPCR. I've seen a couple of different examples, okay? It can do a number of things, but if you recognize it as just a domino effect, then you can understand how it works, okay? So when the ligand comes and binds, okay, it doesn't have to actually enter the cell in order for something to change on the inside of the cell. Now, once that thing changes on the inside of the cell, you've got those little pieces of the G protein going up here, little pieces going over here, and then they're going to dock to something else, and then we see some sort of change, okay? Now, let's take care of some acronyms. GTP stands for guanine triphosphate, okay? Or maybe like guanylyl triphosphate. What is it? It's guanine, okay, with a ribose and 3 phosphates. GDP, you guessed it, guanine with a ribose, 2 phosphates. GMP, if you ever see that, don't be scared, guanine monophosphate, okay? Now, right now, what I'm seeing is GTP and I'm seeing GDP, okay? Those are the ones that we need to be worried about. And it's just like ATP. That's the easiest way to remember it, is that this is the uncharged version. This is the charged version. If you ever see this, it's really weird, okay? But we have the charged and we have the uncharged, or we have the charged and we have the uncharged, okay? And those can go interact with other things. Now, some of the things that I'm seeing they're interacting with is something called AC. That's called Adenylyl cyclase, okay? And pKa, which I believe we talked about before, which is protein PhosphorylaseKinase A, okay? So remember, if it ever ends in ACE, it's an enzyme that's doing some sort of work, okay? And right, so, and whatever it's working on is also in the title, okay? So adenylyl cyclase is something that is going to turn adenine into a cycle. Does that sound familiar from anything we talked about? That's right. CAMP, cyclic AMP, is formed from ATP into cyclic AMP by adenylyl cyclase, which is abbreviated in a lot of diagrams as AC. Okay? And that can be initiated by G proteins. Typically, from what I saw, I think it was the beta and the gamma component of the G protein are going to bind to the adenylyl cyclase, which is then going to convert ATP into CAMP. Now, okay, so what's the point? Okay, this is converting this into this so that it can go and touch this, so it converts this into this, and then converts this into this, and then converts. What's the end game here? Why do we have so many freaking steps? Well, let me remind you, we have so many steps, and we have all these little middlemen going on inside because this is evolution. Evolution does not take the direct route. Evolution just builds on what was there before. So that might mean that you have 6 steps in a process that seemingly could have been redesigned as a 1-step process, okay? So you can look back at bacteria, or you could look at other type of life forms or other simpler mechanisms of signaling and see, oh, it probably just takes 1 step or 1-2 steps. And then you look at more complex areas, probably like our brain and, other areas of like higher evolved eukaryotes, and you could see, oh, it's a 6-step process, but you could probably see a similar 1 or 2-step or 3-step process in bacteria or in other prokaryotes, okay? All right, hopefully that helps. Maybe I got through it all. That looks like most of my notes are pretty much covered. So, 14 minutes, my gosh, this is, I guess it's faster than I do in class kind of, right? Check it.